So here we are on the rocks at Portobello Marine Lab with Gabby here, and Gabby's a PhD student working on, what are you working on, Gabby?
Hi Steve, I am working on the invasive seaweed, Untitled Chapter, and I'm studying about the ecology of it around sites in southern New Zealand.
Okay, so this seaweed, tell us a little bit about where it lives and how it survives.
So Undaria was originally from around the Sea of Japan, but it's come to New Zealand in the Otago Harbour about 30 years ago, and so it lives along here in the rocky shore. You'll see it quite densely sometimes in the intertidal areas, and as well it goes down to 8 meters or so in the subtidal.
So where is it here? Yeah.
So right here we can go out and actually just grab some.
Yeah.
There's a nice mix of other reds and green seaweeds in here, but then Undaria is kind of mixed right in there.
And why do we find this is sort of the active seaweed kind of zone? There's so much seaweed here just at low tide and below low tide.
So there, in this area, the seaweeds are able to utilize the light, and as the tide moves in and out, the fluctuations in the light may vary a little bit, but the intertidal area is really nice because they can get more light at the lower tides, and that just changes throughout the day.
Right. So this is our Undaria.
This is the Undaria, so this is the invasive seaweed.
Yeah.
And so it's really easily identifiable here because it's the only brown seaweed in New Zealand that has this really obvious thick midrib or the stipe, and it has these reproductive parts. They kind of look a bit like chips at the base, so that's actually the spores on it is where it reproduces, and it's attached here at the holdfast. And so it'll just be sitting like this in the rocks, and it's an annual species, so it'll grow mostly in the winter and throughout the spring, and then start dying back in the summer a little bit.
That's a bit unusual, growing in the winter. So.
Yeah, it is. I think I'm not exactly sure why it's a bit opposite maybe than you would expect things growing when there's more light in the summer, but it is more reproductive in the winter and just grows back. Maybe the waters are a bit colder, and that's when it's taking advantage of the nutrients.
So photosynthesis for this plant, it can happen in pretty cold conditions.
Yeah, it's actually pretty surprising. It can happen in really cold conditions, I think. Photosynthesis, it really just relies on the light and the wave energy. You know, the temperature that it lives in is different parts of its, I guess, way that it photosynthesizes.
Right. And so you said down to 8 meters.
Yeah.
So down there, what sort of light's it getting down there?
It can be really dark down there. Usually you're seeing more like red seaweeds down there that can take advantage of that blue and green light. Yeah. But yeah, some of it, you'll see Undaria down there, and other different kinds of seaweeds that can photosynthesize even when the light levels aren't as high.
So it sounds like it's a pretty efficient thing. So why the interest in this invasive seaweed?
So we're interested in it because we don't really know what it's doing to the native seaweeds here in New Zealand, and so we're trying to learn a little bit more about that, seeing how it's become such a dominant part in lots of the intertidal areas. We just want to know a little bit more about it, and since there's so much of it, we have a lot of samples that we can look at and utilize.
Right, and what are the areas you're mostly working in?
So I'm doing some work in the East Otago Taipere, which is just north of the Otago Harbour, and I also have some sites in Stewart Island and Fiordland and other places in southern New Zealand.
Right, and so can you take us through, like, what's a day in the field like for you? What do you have to deal with?
Yeah, so we are scuba diving or snorkeling a lot for my work to collect samples or to remove the Undaria out of the water to look at it and the native species. So it will involve a lot of prep and planning to just get all the dive equipment and plans squared away, make sure we have skippers and crew and everyone knowing what we're doing. And then a lot of checking the weather and seeing, making sure it's a good day to go out. We need to have waves that aren't too big. So like this is a pretty nice calm day here in the harbor. We could have teams out here working and surveying, and also making sure that the visibility in the water is pretty good, because when you're scuba diving you want to make sure you can see your dive buddy, or you can see the seaweed that you're actually looking for, hopefully. And
I understand you also take before and after photos of a lot of the sites.
Yeah, so we'll take before photos when we'll see an area that has a lot of Undaria that we're going to be taking out for the experiment, and then taking a photo afterwards to see the difference in it and all the natives that are kind of there that can now receive all the light to photosynthesize.
Right. It's amazing because our topic has been light, and we've looked at waves and we've looked at the nature of light, and then we've looked at the energy transference, you know, things like vision and photosynthesis, and then also transformation, you know, photography and images and photochemistry. So it seems like your study and your work touches upon aspects of all of these things.
Yeah, and I really love studying seaweeds because, you know, they're the foundation and the building blocks of life. They take that light and that energy and turn it into something useful, some oxygen and nutrients for the other organisms in the sea to utilize. And it's just a really cool process. They can take the carbon dioxide out of the water while they're photosynthesizing and sequester that carbon, and it's all just, it's simple processes, but really amazing processes too.
I can see you're quite enthusiastic and excited about this. There's another name for it, isn't it? I think, is it wakame?
Yeah, so wakame, you'll see it, maybe you've even eaten it before in a seaweed salad of some sort. When it gets cooked or eaten, wakame turns bright green, so it'll be almost as green as this sea lettuce that you see here. But
you could collect it and make a nice soup out of it?
Yeah, definitely.
Do you?
I've tried it before, yeah. So these parts, the leafy parts, are really nice and they're edible. And you can even do those lower sporophyll parts. You can cut those off and cook them, and they're really nice. I like putting it with sesame oil myself.
But like a miso soup.
Yep.
All right.
Exactly.
So if it's an invasive seaweed, maybe it's, if you can't beat it, eat it.
Yeah, definitely. That could be one of the things we can do. It's also useful as like a fertilizer as well. So you can even just dry it and add it to your gardens. And nice compost addition for some nitrogen there.
Okay, and being, you know, down to 8 meters and coastal, is a rough day with a bit of sediment a problem for it and for you to work?
For us to work, so sometimes if there's a lot of sediment in the water, it can be quite dark maybe, or it can be hard to see. You know, I've gone diving before, and if you can only see just a bit in front of your face and you're trying to look for your sights, it's really hard to see. So the sediment isn't an ideal situation for us when we're working, and then also it kind of blocks out some of the light even. So for all the seaweeds that are in that understory of the kelp forest there, it can be hard for them to photosynthesize.
Right. Hey, thanks very much, Gabby.
Yeah, no, this was great, Steve. Thank you, that was really fun.
And so when are you next out in the field?
So I'm going to be going to Stewart Island next week, actually. So I'll be going out there in the field and looking at some of my sites on the southern side of Stewart Island.
Oh, best of luck, and hope the weather's fine and you don't have any big swells or waves to deal with.
I sure hope so too. Thank you.
Thanks, Gabby. Okay, we've been studying light, particularly in relationship to energy movement and energy transference. And we've been talking to Gabby about her work with the photosynthesis of Ondaria, and here we are in the algacultural lab at the Portobello Marine Lab, and here's Liz, who works here with, of all things, algae. Liz, would you like to tell us a little bit about the world of algae?
Sure. I've been working with algae for over 6 years, and to understand where it lies within the marine ecosystem and also within aquaculture, which is where I have been working, is to say that it's on the first level of the trophic levels, and it provides food for juvenile shellfish. If we want to spawn salmon, oysters, any kind of shellfish or finfish, we start with providing them with food that they would normally get in their environment. The food that we produce here is really high concentrations of it. If we were in the wild, it would be much less apparent. You wouldn't necessarily see what the animals are feeding on. You do, however, get algal blooms. You hear about it in the news. You can get harmful algal blooms, or you can get good algal blooms, which is basically what we're creating in here. In an algae lab, you are generally working indoors, and that is because you're trying to control the environment. And with that, it's simply so that we have better success. If we did this outside, you have variability from light, climatic conditions, temperature, and that goes through the year. So come into my lab.
Whoa, the lolly shop.
Yeah, it does look a lot like a lolly shop. So at the moment the lights are off. It's daytime, but I just turn the lights off, so I work in here during the day. But this gives you an understanding of the light controlling the growth of these cultures. So we're on a 12-hour cycle. We have 12 hours of light on a timer, and then at night time the light goes off. So at the moment, these cultures are growing in white light, and they also get a little bit of daylight coming through. That can interfere again with the growth. It may spur growth on, which, you know, if we didn't have windows, this lab would be even more controlled. We have a temperature control system. So this is currently set at 15 degrees, which is reasonably low, but we're in Dunedin, and so the ambient water temperature outside today, I think is 12. So during the year, you'll get a variation in seawater temperature and in your air temperature, and that is also going to have some effect on how the success that you have in growing your cultures and what types, what species of cultures you're going to get.
So these species here, you've got some green, you've got some brown ones,
yep.
And I guess we're familiar with the green being chlorophyll. Is that?
Yeah.
And the browns have got those xanthophylls, chlorophylls.
Mm-hmm. A variation of, so depending on the species that you've got growing is going to depend on the action or activity of the chloroplasts.
So besides the light, what else do you need to provide them for this photosynthesis?
So into what we would call a closed system, which I just happen to have one cooked over here. So I should explain that actually it is literally cooked.
Yeah.
Again, to control the environment, we sterilise water in an autoclave, and this water has been sterilised, autoclave bag. So this is 35 ppt salinity seawater. I've put nutrients in there, which is just a whole load of food, I guess is the easiest way to describe it. And depending on what I'm growing, if I'm growing diatoms, which again is single cell, quite often generally browns or reds, they require sodium metasilicate, so I'd also put some of that in there. So in this controlled environment, this seawater has just been cooked, and what will happen is that I will put an algal culture, a green or a brown, which we have on site at the moment. I'll inoculate different flasks in different sizes. So I will put some of this culture into here. It will grow out maybe 5 days. So I'm just basically increasing the cell size, cell density. This flask is again seawater with nutrients and maybe some silicate. So this will go into here and it will grow for a few days, and then I'll upsize again to either this size, which is 500 mils. I might go up to a litre, depending on the cell density of the culture, and then I will eventually go through to a 5 litre. And with that 5 litres, which is now up to about here with algae and really good cell density, I will inoculate that into these carboys. A better example actually is these carboys, which again have 35 ppt salinity, they have nutrients, they have metasilicate, and I will inoculate those, and then I'll put them onto these filters, which will maintain a clean environment of air coming through, and we also have a mixture of CO2, carbon dioxide, coming into that system.
So we've got the carbon dioxide, we've got water, we've got the nutrients, and then the photon energy coming off the lights kicks it all off. In the chloroplasts, and away we go.
Yeah.
And as you said, I think in the introduction, this is a lab constructed bloom, a bloom. Yeah. So what's the end result? What's the purpose of this?
So for what we do here at Portobello Marine Laboratory is a lot of research for marine scientists, marine species. So I describe my job growing algae very similar to I'm growing grass for sheep. So if you're on the farm and you're growing sheep or cattle, you need to provide a good food stock or food supply. And part of that food supply is about giving the animals something really nutritious and with the kind of components that you want them to eat and improve certain qualities of their physiology. So I'm growing grass for sheep here, or basically I'm growing a green algae for the first stage of the primary production, and this will be fed to newly spawned spat for shellfish and finfish, and it will continue to feed them up until they go onto a different diet. The different colours or different species, that's about how nutritious the food is. So this green is a very general and fast-growing, easy to grow species. Browns are a little bit trickier sometimes, but browns are actually more nutritious. So, and it's about the fatty acid profiles that are within the cells, and that again comes down to photosynthesis and light waves, etc.
So we hear about this, don't we? You know, omega-3s, omega-6s, this sort of thing. You know, it's supposed to be a good thing about in fish as part of our fish diet, but they get it. They have to get it from
here.
From here.
Yeah.
Okay.
So in actual fact, that's it. You know, eat salmon, get your omega fatty acids, etc., or your omegas, and it turns out that it actually kicks off in algae. So algae is actually vitally important for what's going on out there in our marine environment as far as kicking off, you know, the primary production. It's not really recognised by a lot of people because these are single cells. If you see these in the water, if you're swimming in the water, you're never going to see them, as opposed to seaweed, which is a macro or large algae. So they're all kind of like within the same evolutionary system, but these guys are single cell. They're available to animals or spat with tiny little mouthpieces or filter feeders, so they can just filter this through, and that's where they're getting their nutrition from.
Particularly important for plankton, I guess.
Yeah, well, so this is phytoplankton, as in phytoplant and minuscule, and then you get the rotifers and the other really tiny organisms that make up like zooplankton and stuff like that. So I find algae interesting because it's a quiet little achiever. Yeah, just kicks away, and it's this is a nurturing sort of a job. It's partial gardening, it's partial baking, and it's nice just to see the cultures developing. So I've been gardening for quite a few years, and I've been gardening, had been growing algae. I see it quite similar in a way. This is like growing orchids. It's probably easier, but once you understand the environment that you want to grow in, you can, you know, and the nutrients that are required, and the amount of light that comes into the system, then you can start getting some pretty good results. Yeah.
Hey, thanks, Liz. That's absolutely fascinating.